peptide bond forms when the acid of one amino acid joins to the amino of another amino acid. In this example, the acid group of glycine joins with the amino group of alanine. The result is glycoalanine. The acid group of glycine loses an atom of hydrogen and an atom of oxygen. The amino group of alanine loses an atom of hydrogen. These atoms combine to form a water molecule. More peptide bonds can form in the same way, creating a chain of amino acids called a polypeptide.
Okay, so again, same sort of, right, dehydration, synthesis, reaction. I know that's not a vocab word, but it's like it's just, you guys want to start looking for patterns, and obviously this is a pattern. These monomers, these individual units are linking up to make long molecules of repeating, repeating units, and losing a water to do that. Okay, now, proteins are what build us, and they're what they also send messages in our body. They also help us with all the reactions that happen in our body. So there's just tons of them, and they have all different function. And what's really interesting about them is their function is completely related to their shape, form, and function. I've mentioned this theme also. And so right away there's actually 4 different types of structures, simplest to more complex with proteins. So the primary structure, a vocab word, right, you guys? Tyler, L, you with me, man? You okay? So it's just a polypeptide chain. So this is the simplest type of protein you're going to get. It's just a chain. Primary meaning first, right? So the simplest. Now, secondary structure in proteins get a little more complicated. They're not just a chain, they're a chain that's folded or is spiraling. And you can see it's just a little more complex. And it does this almost always through hydrogen bonds. Remember your water molecule? Do you remember it's polar and opposites attract? Do you guys remember this? So the way this thing gets folded is there's going to be some hydrogen bonds across holding the shape in place. And then tertiary structure is just a protein that's even more complex and has a combination of things. Maybe has a helix, maybe has a pleated area. It's also, you could see that shape, it's folded like that. It's going to stay like that. That's its shape. It won't work if it loses its shape. Kind of like a lock and key. Now, I just realized something. I got a little confused, and you guys don't have enzymes in this chapter, do you? Okay, so I will rework that question. I only had about 3 people up to last night even try and tackle my enzyme question, which enzymes are proteins, so that's why I started talking about that lock and key analogy. So I'll revamp that question if you guys are still struggling with what you want to talk about in your groups, because I'm not seeing much activity at all. And I want to remind you all that is a grade. It'll be worth 100. If I don't see activity before the exam, you will have a 0 for not posting anything on Edmodo in your small group. And you had to post 1 question, and then you had to try and respond. And the idea here is I have 4 of you, talking back and forth, and making each other think, making each other learn things. So I need to see that if you want your 100 points. Okay, so back to protein structure. This one, again, it's folded in a more complex manner. So it's like not primary, not secondary, it's tertiary. It's the next complex way that proteins are structured. That was just talking also about the folding, like I talked about, could come from hydrogen bonds. It could also come from, some areas could be nonpolar, some areas could be polar, so they're attracted to each other or they're trying to stay away from each other. There's lots of interactions to making these shapes. The last structure is quaternary and a 4th, right, quadrant. And what that means is there's more than one chain even involved in this protein. Okay, so there's, there could be I don't know. I know of many, like this one has 5 different individual protein chains, polypeptides, that are then folded together. We're going to work with those enzymes next week when we get to that lab. Okay, then nucleic acids. And I think I mentioned this before, or maybe I'm getting confused with my other class. I want you to memorize it like right now. This NA, there's 2 types of nucleic acids. What are we talking about? Just yell it out. Nice, wonderful. So DNA deoxyribose nucleic acid, and then ribonucleic acid. I have to drink more water. Okay, so this is where they're getting their names. Deoxyribose, do you see that right there? That's the name of the sugar that's in DNA. And it has to do with one less oxygen, and ribose is the one in RNA. So their monomer of the DNA is called a nucleotide. Am I going to get a picture? Good. So here, see, we have, these are the 2 different sugars. You can see how they vary. Again, you don't need to remember the structure of that sugar. In DNA, there's 4 different nucleotides, A, T, C, and G. And notice the T and the G are a little bit larger than the nucleotides of the C and the A. And you should be aware by now that C's always bond to what? G's. And A's always bond to T's. And again, we'll spend more time with that. Okay, so I've mentioned that already. I do want to show you our DNA model. So you could see the repeating units. You could see the 4 different colors. And you could even see its alpha helix. It's spiraling, how it's spiraling. So the nucleotides are right here. I have a purple phosphate, an orange sugar, and then my nitrogenous base, which are the ones I just showed you. And then another picture of each of the nucleotides. Again, the sugar, the phosphate, and the nitrogenous base. And I mentioned how RNA is different in the way of the sugar, but it also doesn't make a double strand. It doesn't do that helix. It's just a single strand, and it's really made off of DNA. And we'll talk about that much later. So Watson and Crick get full credit, basically, for coming up with the model, the shape of DNA, even though they had lots of help. And there's some interesting stories with it. Rosalind Franklin here and Maurice Wilkins, to be really controversial, some people believe, and there's evidence to this, but there's a little bit of contradictory to that. It's not so harsh. Wilkins was working a little bit with Watson and Crick, was at the same school as Rosalind Franklin. She has famous photographs. She was figuring out crystallography, X-rays of molecules, which we still use today. And Wilkins basically slid, supposedly, Rosalind Franklin's picture over to Watson and Crick. And when they saw, and they do acknowledge that when they saw her X-rays, pictures, that they were able to put the puzzle together. But you never hear about Rosalind Franklin. Has anybody heard about Rosalind before? Yeah, good, a couple. And now she's making it into the books, like she's in your book. And pretty interesting. She got really frustrated with the whole thing. Not just that, but being a woman in science during those, in the 40s. She ended up just getting out of the nucleic acid business and went to the protein business and did really, really well and ended up winning prizes. But also because of working with the X-rays, she ended up dying of cancer as well. Kind of like Marie Curie. You guys know Marie Curie's story, and we'll talk more about that one. So real quick, here's your DNA.
The DNA double helix is composed of 2 long chains of nucleotides. The backbone of each chain, shown as ribbons, is composed of alternating sugar and phosphate molecules that connect the nucleotides. The two chains are held together by specific patterns of hydrogen bonding between pairs of bases, A to T and G to C, and the chains coil around a central axis to form a double helix. The diameter of the double helix is equal along its length because a double ring A or G base called purines always pairs with a single ring T or C base called pyrimidines. There are 10 nucleotides per turn of the helix. Only a small portion of a model of a DNA molecule is shown in this example.
Is that it? Yay, you made it.